This is a production of Cornell University.
Yeah, I'm Will. Today I'll be talking about trying to understand how environmental factors influence the contamination of maize by mycotoxins in a very diverse smallholder farming system in Tanzania. So just some background about why mycotoxins are a risk. These are secondary metabolites produced by crop fungal pathogens. In the tropical lowlands, maize is primarily affected by two classes of mycotoxins, aflatoxins and fumonisins. Very different chemical structures, but both are toxic to both human and animal health. Both cause ear rots. We have Aspergillus ear rot and Fusarium ear rot illustrated in extreme cases here. And aflatoxins are more common at extremely hot temperatures, while fumonisins slightly more moderate, but there's significant overlap. And aflatoxins are more toxic with regulatory limits that are, excuse me, orders of magnitude lower than for fumonisин. So from a landscape-wide scale, these toxins are highly influenced by the environment. In a nice study done by Laura Smith and Samuel Matiga from our lab, they looked at eastern and western Kenya, where there's sort of more rain versus semi-arid areas in eastern Kenya, and found that here in the semi-arid areas, there's more aflatoxin, while in the rainier areas, there's less aflatoxin. And it's not shown here, but for fumonisин, the relationship was reversed, with more fumonisин here and less in eastern Kenya. So this kind of gives you a sense that there are spatial dynamics that are related to the environment for the production of each of these toxins. And when we think about what sort of risk factors, environmental risk factors there are for mycotoxin contamination in maize, you really have to contextualize it within the maize growing season. So infection of maize ear rots really occurs during the reproductive stages. Spores of the fungus infect either via silks or via some sort of injury to the ear. And so a stressed plant during this stage will be more susceptible to infection and subsequent contamination. So if a plant experiences drought during this time, then it is more likely to have mycotoxin at harvest. Whereas later in the season, after fungal establishment on the ear, rains at maturity are actually more likely to make a more conducive environment for fungal growth and result in more contamination. Across the season, soil health is very important, as it's poor soil health, such as low nitrogen, low carbon, contributes to plant stress and makes a plant more susceptible to infection. So I'm interested in how all of these environmental factors could be used to model mycotoxin contamination across a region. And so the study site is Kongwa District here in central Tanzania. And Kongwa District is a semi-arid smallholder farming system. We have collaborators working on a childhood nutrition trial there. And as part of that work, we've established this contamination by both aflatoxin and fumonisин. And so I'm just showing here that childhood stunting, which is what this trial is looking at, is elevated in Kongwa and across all of Tanzania. So this is especially relevant in these areas, as even earlier this year, high aflatoxin contamination of maize has been reported to cause an outbreak of aflatoxicosis, which resulted in 8 deaths in regions bordering Kongwa. So to try and get at this question of how the environment's influencing the geographic dispersion of mycotoxins across a smallholder farming system, I first went to Kongwa, interviewed farmers, and sampled maize at grain mills, then quantified samples maize and aflatoxin values by an enzyme-linked immunosorbent assay. Next, acquired remote sensing imagery of different environmental risk factors, and finally tried to model mycotoxins as a function of these remote sensing indicators, so concentration of mycotoxins as a function of things like NDVI, soil carbon, harvest rate, et cetera. So for my survey, I visited Kongwa District, and over the course of 17 days, visited 88 grain mills and interviewed 306 customers of these grain mills and took a sample of maize from each one. These grain mills are sort of local nodes of maize processing. So everyone within a maybe 5-kilometer radius of that grain mill goes there to have their flour or their whole maize ground into flour for household use. So it allows you to understand local variants in mycotoxins as well as sort of georeference that so you can look at environmental indicators. So from a questionnaire, here's my research assistant, John Mshanga, talking to a customer outside of a grain mill. We learned many things, but just a few highlights was that planting date was generally December to January. Harvests were mostly in May. I was there late May, early June, and people would sort of harvest portions of their field at a time. So it wasn't a single date for all of the harvest. And all but one respondent said that they experienced drought this year. And when they experienced drought was mostly in the middle or middle end of the growing season, which correlates to those risk factors that other papers have established. And when I did my ELISA assays and looked at mycotoxin contamination within my samples, here is log transformed aflatoxin. This bold red line is the legal limit of 10 parts per billion. And you can see that a vast majority of samples exceed the legal limit, and many of them by orders of magnitude over the legal limit, just showing how extreme the contamination by aflatoxins were this season. Fumonisin contamination relative to the legal limit was not quite as severe. So in terms of looking at growing season indicators of crop health, I used normalized difference vegetation index from the eModus Aqua satellite. And hopefully what you can get from this GIF is that across a growing season, there's both temporal and spatial variation in NDVI values. And I should mention that each sample had a NDVI averaged over sort of the distance someone walked to the grain mill. So I knew their farm was X kilometers away, but I didn't know in which direction. So it's for someone 2 kilometers away, it would be a relatively smaller radius versus 8 kilometers would be a larger radius of average values. And you can also use NDVI to compare to previous years with this data set. And so here at 100% is the median NDVI of approximately 20 years of data. And you can see that for the beginning of the growing season, things were more or less at the median. Then after that drought began in the middle of the season, you can see that compared to past years, NDVI plummeted, and then rains started again late in the season, and you see a sort of rebound effect. And I want to show sort of how this correlates to our thoughts of drought affecting during the reproductive growth period, and then rains occurring again at the end of the growing season. I also looked at soil health using the Soil Grids 250-meter data set. This has many soil attributes at high spatial resolution. And in particular, we looked at organic carbon stock, which is the amount of carbon within a given area. And see some greener areas. These are like mountains where there's actually no farming and there's more trees. But across mostly these white and beige areas that are very low values are where most of the farming is going on. And so that sort of is how I'm trying to monitor this other risk factor of soil health. So I use several different models to try and model both aflatoxins and fumonisins. Only models for aflatoxin were significant based on environmental factors. I ended up choosing a mixed effects model incorporating both fixed and random effects. A reduced model found that the fixed effects of NDVI in April, May, and organic carbon stock from the 15 to 30 centimeter root zone, along with the random effects of each grain mill. So samples were associated with the grain mill, and I used that as my random effects. In doing this, I want to focus first on the estimates for my fixed effects. So in April, so nearing the end of the grain The growing season, there's a negative association between aflatoxin and NDVI, sort of as we'd expect. The more stressed crops were negatively associated with aflatoxin, whereas in May, after the growing season, there was a positive association shown here, sort of maybe suggesting that areas that had more rain at the end of the growing season were more likely to be contaminated. And as other studies have shown, organic carbon was negatively associated with aflatoxin. So in total, the fixed effects explained 11.2% of the variance in aflatoxin, which is not a huge number, but for such a small area and a diverse system, it is significant. And when I account for my random effects, along with fixed effects and conditional R-squared, it's almost 28%. I wanted to see how well this worked for predicting if I held samples out of my training set. So doing that, I could compare predicted and observed aflatoxins in my test set and found a strong correlation between predicted and observed. Though I should note that because of the low variance explained, it's a pretty narrow range of predicted values. So takeaways from this are that Congo District in 2019 had an extremely tough maize growing season that resulted in significant aflatoxin contamination. However, it did provide an opportunity to really understand how these environmental risk factors that are freely available and are pre-harvest could be used to create predictive models. Obviously, such models would need further validation across locations and years, but could help plant breeders understand variance in mycotoxin contamination across regions, or even public health experts who want early warning systems for these types of problems. With that, I really want to thank many people, including everyone in the Nelson Lab, our collaborators with the mycotoxin mitigation trial I mentioned, John Mchanga, my research assistant, all the participants and people in Congo who made this much easier than it could have been, and funding from the Atkinson Center and the Einaudi Center. So thank you, and I'd be happy to take questions.
We have time for a few questions for Will.
Yeah.
You are addressing actually pre-harvest infections,
Yes.
but I think that the post-harvest infection is another a major actually sort of issue there too. Right?
Yeah.
So,
yeah, you're very correct. So he asked about the difference between pre-harvest and post-harvest contamination. Aflatoxins are notorious for especially post-harvest problems, and there's a lot of research on storage techniques and processing techniques to reduce that. I wanted to just focus on the pre-harvest aspect, and so actually that's why I was, when I visited, it was directly after harvest, so samples were only days or weeks from being in the field. So I want to sort of isolate that part, and then understanding post-harvest issues is a whole nother part of it, but yeah. Judy?
So I assume that environmental factors and storage factors have probably the biggest impact on aflatoxin. But that said, what do you know about the genetics of what the farmers are growing? Is it kind of a uniform genetic stock? Or?
Yeah, so I did in my questionnaire have questions about what seeds people were growing and all those kinds of things, and there's first very low understanding of what variety. I mean, people are growing open pollinated, saved seeds. This is, I think I met one person out of the over 300 who had purchased hybrid seed or something like that. So there are no inputs. So very diverse genetics. And so that was something I was interested in trying to parse out, but was ultimately kind of impossible. Yeah, but genetics are highly quantitative, low heritability for this, but interested in trying to look at that a little more too. Yeah, Leah.
Am I right to say that one of the people you interviewed said that they experienced droughts in the seasons, right?
One said they did not. So 305 out of 306. So that was like a very unanimous, like loud signal to me that it was a big problem. Mark.
Well, are there cultural practices or fungicides that they can use to minimize the problems with aflatoxin?
In terms of cultural practices, a lot of it has to do with, I mean, some say early maturity varieties, so they get out of the field quicker, proper drying techniques, there proper storage techniques, so even some trying to use anoxic storage conditions, dry storage conditions. This is really like an issue that involves so many different factors, even sorting post-harvest. But in these very, very low input systems, it's difficult to say there's exactly one thing that can solve it all. Yeah.
All right, let's thank Will.
This has been a production of Cornell University, on the web at cornell.edu.